Just to save time for all the science, we'll just jump right in with our first speaker, Benjamin Kroeger.
Okay, thank you. Thanks very much for the opportunity to speak here today. I'm Ben from Clive Wilson's lab at the University of Oxford, where we're studying exosome biogenesis and secretion using Drosophila secondary cells. We study secondary cells which are found within the male Drosophila accessory glands, which you'll see here. This gland consists of a secretory monolayered epithelium of two cell types, main and secondary cells. The secondary cells are clustered here at the distal tips of those glands, and we visualize them by expressing a human exosome marker called CD63 tagged with GFP within those cells. In a previous publication by our group, we showed that secondary cells secrete exosomes into the seminal fluid that you can see over here, and these exosomes are passed on to the female during mating, where they interact with her reproductive tract and also, interestingly, fuse to sperm in a manner analogous to how exosomes from the prostate do. They've been shown also to carry functional effects in regulating the female post-mating response. We're using secondary cells as a novel in vivo model of exosome biogenesis, and for those of you who are not aware, exosomes are thought to originate classically within late endosomal multivesicular bodies. These compartments often carry on their surface a small GTPase called Rab11. They're born through the activity of large protein complexes called the ESCRT complexes that bud off intraluminal vesicles into that compartment. That compartment then has the chance to either fuse to the lysosome and be degraded or be secreted by fusion to the plasma membrane. This releases the intraluminal vesicles as exosomes. The exosomes themselves carry a whole host of different molecules, including microRNAs, receptors, mRNAs, you name it, they have it. And these molecules enable them to carry out their particular functions in target cells. They've been shown to have important functions in a variety of biological contexts, including cancer, immunity, development, and in our lab, reproduction. However, despite these important roles, some key and fundamental questions are not well understood. For example, how is that heterogeneity in exosome cargoes actually being generated when all these cargoes are going through the same type of subcellular compartment? So we're trying to study these type questions using secondary cells, and we're able to visualize the endolysosomal system of secondary cells by expressing this human exosome marker. And as you can visualize here, it localizes on a variety of different compartments within that cell, both acidic and non-acidic compartments. You can see the acidic ones are co-stained with this acidophilic dye lysotracker red. We've been able to identify the identity of these compartments by using the small Rab GTPases, and here I'm showing you the gene traps of Rab7YFP, which localizes clearly on the limiting membranes of acidic compartments within these cells, labeling them more as late endosomal or lysosomal type compartments. We've also observed that these non-acidic compartments often carry the Rab GTPase Rab11 on their surface, which places them more as recycling type compartments, or as we've seen at other points in this conference, they could be actually exocytic type compartments. Why we think these cells are wonderful models of exosome biogenesis is because of the size of their compartments. For example, here, if we compare a PC3 prostate cell, in which the multivesicular bodies or lysosomes are labeled with red, you can see that the size is just almost 10,000 times as big, enabling us to visualize their contents in far greater detail. However, we want to go further than this and to actually visualize intraluminal vesicles within these compartments, and to do that, we've utilized super-resolution light microscopy. And here I'm showing you a super-resolution 3D reconstruction of an individual secondary cell in which we've lit up all of the subcellular, well, most of the subcellular compartments using CD63. And as you can see, as I'll play this movie, you can observe GFP-positive puncta within these large acidic compartments, as well as you'll notice that the lysotracker staining, in fact, looks more to be in microdomain type structures within this compartment. We've actually identified using antibody stainings to GFP that this compartment is in fact filled with CD63. However, the GFP is in fact quenched within those compartments. You'll also notice here some really complex membranous materials within the non-acidic compartments. And we can look at this even clearer by just looking at a single optical slice through one of these cells. And we were really excited because this was for the first time in a living cell we were able to visualize intraluminal vesicles of exosome size within these appropriate types of compartments where they're thought to originate. However, we were even more excited because we could observe intraluminal vesicles carrying exosome cargoes in these non-acidic compartments too, which, if you'll remember, were Rab11 positive. We can look at some of these compartments blown up here, and you'll observe that the diversity and abundance of intraluminal vesicles in these non-acidic compartments. And despite some of them being quite big, you'll note there, most of them in fact fit into the classic exosome size range. We've utilized time-lapse microscopy to show that these compartments are secreted from the cell. And here's a movie. If you follow this compartment here, you'll observe it fuse to the apical plasma membrane of the cell and release GFP into the lumen. Next, we wondered, are there in fact some compartment-specific exosome cargoes that could help us to explain heterogeneity seen in exosomes? And in fact, we did a screen of various molecules and found that the FGF receptor, which is endogenously expressed by secondary cells and is secreted into the lumen at very high levels, localizes quite particularly on a subset of intraluminal vesicles. For example, you can see here very beautifully inside these non-acidic compartments. This is again super resolution microscopy. However, in contrast to the CD63 I showed you earlier, it's far less enriched within these acidic compartments and mostly excluded from the limiting membranes of those compartments. Data from this and other markers, which are quite specific to particular compartments, have suggested that in fact these non-acidic compartments are the main sources of exosomes within our cells. We've also identified that the small GTPases could be interesting markers of exosomes. For example, here where we've expressed the Rab11 YFP gene trap and found that it localizes onto intraluminal vesicles within the type of compartment it normally localizes on. And indeed, we've also observed similar things with Rab7, which localizes on acidic compartments, is also incorporated into intraluminal vesicles there. So these data have led us to develop a new model of exosome biogenesis, whereby exosome cargoes can be selectively loaded in ILVs of specific compartment subtypes. We've observed that probably most secondary cell exosomes are released from Rab11 positive compartments, and that the Rab GTPases may in fact be used as indicators of the intracellular origins of exosomes. We've worked collaboratively with another group in Oxford led by Deborah Gobodan to show that these processes are in fact conserved in human cancer cells, and in those cells are regulated by specific cellular stresses. In the secondary cell model, we've decided to ask some other questions. For example, how are these alternative intraluminal vesicle populations actually being formed? And some key candidate molecules for these are the ESCRT complexes, which, as I stated before, act together to produce intraluminal vesicles within compartments by recognizing and clustering particular cargoes, as well as helping the budding of a vesicle and scission of it within that compartment. However, there have been proposals of ESCRT-independent mechanisms to form intraluminal vesicles, so we wanted to test whether the ESCRTs were actually functioning. And if you look here at a control cell in which, once again, we're expressing CD63 GFP, you can observe these non-acidic compartments are filled with membranous structures, whereas when you knock down the ESCRT complexes, for example, this component of ESCRT1 called VPS28, we observe an inhibition of these intraluminal vesicle formation in these compartments. These data have been significantly affected across a variety of different knockdowns that we have undertaken, and we've gone a bit further than this, and we've observed, for example, in this cell, we've seen that the lysa tracker staining is in fact quite affected, and this led us to the possibility that the ESCRT proteins or intraluminal vesicles were critical in the maturation of these acidic compartments. And we have some descriptive evidence that might help support this hypothesis. So if you look at this compartment over here, you can observe a small intraluminal vesicle that's surrounded by a microdomain of lysa tracker staining. And we've proposed that this might be formed through the activity of the VATPase on these intraluminal vesicles and maintained through the activity of nutrient transporters. This is supported by the finding that this VATPase subunit, VHA55 GFP gene trap, in fact localizes onto intraluminal vesicles where it may be able to carry out these functions. But we wanted to analyze these more quantitatively, and so we've utilized a ratiometric dye called lysosensor yellow-blue. And in short, how this works is if it's more yellow, that means it's more acidic, and if it's more blue, it's less acidic. And if you compare this control cell here, in which we can observe really large yellow late endosomes, to ones in which we have knocked down the VATPase subunits VHA-SFD or VHA-101, you can observe that the pH of these compartments is dramatically affected. And indeed, when we knock down the ESCRT complex 3 member, shrub, we observe a very similar phenotype, suggesting that intraluminal vesicles are critical for the maturation of these compartments. So in summary, I'd like to propose that exosomes are derived from multiple compartments in both flies and humans, that cargoes can be selectively loaded into ILVs within different compartments. The Rab family GTPases can help us identify exosome origins. Alternative exosomes are also regulated by some of the same molecules, the ESCRTs. And intraluminal vesicles can play important subcompartmental functions, for example, in acidification, and also we found in dense core granule formation, but I haven't had time to share that with you today. And with that, I'd like to thank all the people that have made this project possible, my supervisor, Clive Wilson, as well as collaborators Felix Castellanos, Irina Stefana, and an undergraduate student who worked with me, Rosie Marshall. Also our collaborators in Deborah Gobodan's group, including Sumeth Pereira and Xiong Fan. I'd also like to thank the imaging suite at Micron in Oxford, who've helped make the super resolution possible. And lastly, my sponsors who've funded my PhD and travel here to the US. Thank you for listening. I'll be happy for any questions.
We have time for questions.
I'll start with one question.
Yes?
The Rabs, are they just markers, or are they also necessary for the fusion event with the plasma membrane?
So in our hands, when we've knocked down the Rabs, these compartments don't mature properly. So, yeah. No, yeah.
In some of your images, it appears as if Rab11 was actually not on the outside of the vesicles, but rather inside. Do you have any idea how by which process they were localized internally?
Internally?
Well, basically normally you would expect them to be on the limiting membrane.
Yes,
but
that's
in some
right.
of your images, it appeared that they were actually inside the vesicle. So do you know how they get in there?
So in our model, the molecules that are on this limiting membrane get invaginated in and are found within the intraluminal vesicle. I'm sorry, does that answer your question?
No,
Okay.
because it was filled. So you had these intraluminal vesicles,
Yeah.
and within these intraluminal vesicles, they were filled with Rab11, and I was just wondering how this got in there.
So I think that some of our Rab gene traps are also, we find that the Rabs can be found also within the cytosol at lower levels, but they're enriched on the limiting membranes. So it could be that they are just found within because they're not actually associated at that point.
any idea that what are the cargoes of these exosomes?
So that's a good question, and we have found that some molecules using antibody stainings, but we don't know yet which are the molecules that are actually functioning in the female to cause the post-mating response. So in short, we do have some key candidates, but we don't know which ones are totally functioning in the female.
Thank you.
All right, thank you.